Every single one of your cells is running a silent, continuous audit right now. It's not looking at your thoughts or your feelings. It's looking at voltage, specifically the electrical charge across the inner membranes of your mitochondria, the tiny power plants inside you. It's checking to see if they're holding a specific charge, about 180 millivolts. Any that are failing this voltage test get flagged, isolated, and dismantled. Their parts are stripped down and rebuilt into new functional units. This isn't a special event. It's a background process that runs 24/7. But here's the critical point. There's a condition that accelerates this cellular quality control so dramatically. It's like the difference between a slow, quiet maintenance shift and a full-scale factory overhaul. And that condition isn't a new drug or a supplement. It's simply the absence of food. To understand why, you have to start with a law that governs everything in the universe. Not a suggestion, but a law. The second law of thermodynamics. It states that in any closed system, disorder or entropy always increases. A living cell is a masterpiece of order, an island of improbable structure in a universe that wants to flatten it all out. You have specific concentrations of ions on one side of a membrane versus the other. You have proteins folded into impossibly precise three-dimensional shapes. You have entire organelles positioned just so. This order isn't free. It demands a constant, relentless expenditure of energy. Think about the sodium-potassium pump. It's a molecular machine that sits in your cell membrane, and it alone uses up about 30% of your cell's entire energy budget. It does one thing. It pumps 3 sodium ions out of the cell for every 2 potassium ions it pumps in. That's it. That's the price of maintaining the electrical gradient that allows your neurons to fire and your muscles to contract. And that's just one pump. The entire cell is a sprawling metropolis of such machines, all working against the universal tide of chaos. Proteins are especially vulnerable. They are long chains of amino acids that have to fold into specific configurations to work. At body temperature, the thermal energy alone causes a protein with a few hundred amino acids to jiggle and shift, sampling thousands of incorrect shapes every second. Most of these are corrected instantly. The protein's own inherent stability pulls it back into its proper form. But a fraction of them escape into stable, misfolded states. They become rogue, sticky molecules. If not caught, they start glomming onto other misfolded proteins, forming aggregates that the cell can no longer manage. These are the seeds of dysfunction. Then you have your mitochondria. While they produce the ATP that powers everything, they also leak. They produce reactive oxygen species, unstable molecules that act like sparks from a furnace. These sparks damage the mitochondrial DNA, their membranes, and the very proteins of the electron transport chain that make ATP. Every single ordered structure in your cell is under constant physical attack from the thermodynamic noise of just existing. The cell's only defense is to spend energy to repair or replace. It's a never-ending battle. When food is abundant, the cell runs a dual economy. It builds new structures, new proteins, new membranes, new mitochondria, while running maintenance in the background. The growth signal dominates. It's like a city that keeps building new skyscrapers while only doing the bare minimum to patch potholes. The balance is tilted towards expansion. But then the dynamic shifts. When food stops, when those external building blocks vanish, the cell faces a fundamental resource allocation problem. It can't just run on low power across the board. That would lead to systemic failure. Evolution crafted a more elegant solution. It flips the switch entirely. The cell stops building outward and starts consuming itself from the inside, but not randomly. It begins a precise targeted audit, consuming its own most damaged components first, recycling the molecular debris to keep its most essential systems alive. Growth stops. Repair mode accelerates. The balance inverts. The cell doesn't starve. It self-audits. The cell doesn't starve. It self-audits. This is why the second law is the fundamental reason for autophagy. Entropy isn't some abstract concept for physicists to ponder. It's an active physical force pulling every protein, every membrane, every organelle apart in your body right now as you sit here. Fasting is simply the condition under which your cell mounts its most aggressive, organized response to that force. Now we get to the switch. The shift from growth to repair isn't a smooth dimmer. It's a cliff, a phase transition. Think about water. As you cool it, it doesn't gradually get thicker and thicker until it becomes a solid. It's a liquid, and then at exactly 32 degrees Fahrenheit, it's ice. It's a discontinuous jump. The mTOR system in your cells works exactly like this. mTOR is a master sensor, a protein kinase that monitors nutrient availability. When amino acids are plentiful, mTOR is active, and it puts a firm brake on autophagy. The cell is in build mode. When amino acid levels drop below a certain threshold, this typically takes somewhere between 12 and 16 hours of fasting in humans. Another sensor, AMPK, detects the rising ratio of AMP to ATP, the cell's energy currency running low. AMPK steps in and flips the switch. It inhibits mTOR, and this inhibition isn't partial. It's a threshold. Below that critical point, the brake is on. Autophagy is suppressed. Above it, the brake is released and the recycling machinery initiates across the entire cell. This explains why the difference between 14 hours and 16 hours of fasting isn't just 2 hours of more benefit. It's the difference between almost no autophagic induction and a significant induction. The benefit isn't linear, it's binary. You're either on one side of the phase boundary or the other. A person who eats dinner at 8 p.m. and breakfast at 10 a.m. has fasted for 14 hours. They may have produced minimal autophagy. The person who waits until noon has fasted for 16 hours and has likely crossed the threshold. Those 2 extra hours aren't just additive, they're transformative. They represent crossing from one cellular operating system to another. The man who figured this out wasn't studying fasting at all. He was watching yeast. Dr. Yoshinori Ohsumi was looking at yeast cells under starvation conditions, and he saw something no one had ever bothered to look for. He saw the vacuole, the yeast equivalent of a lysosome, filling up with tiny membrane-bound bodies. He didn't know what they were. So he did a genius thing. He engineered yeast strains that couldn't break down the contents of their vacuoles. When he starved these mutant yeasts, those tiny bodies just accumulated, becoming impossible to ignore. He was looking at autophagosomes, the cellular garbage trucks. He had found the threshold in yeast. Over the next decade, he painstakingly identified the genes responsible, the ATG, autophagy-related genes, and mapped the molecular machinery that builds these autophagosomes, targets the cargo, and delivers it for destruction. The work was done in yeast, a single-celled organism most molecular biologists overlooked, studying a process considered a minor housekeeping function. But the threshold he identified operates in your cells, too. The molecular switch is so fundamental, so critical, that it's been conserved across more than 1 billion years of evolution. The ATG genes in yeast have direct counterparts, homologs, in every cell in your body. The machinery is identical. The trigger is identical. The physics is identical. Ohsumi was awarded the Nobel Prize in Physiology or Medicine in 2016 for describing a process that had been running in every eukaryotic cell since before multicellular life existed, and that nobody had ever bothered to map out with such precision. So what happens after the switch flips? Because the structure the cell builds is a marvel of nanoengineering. Autophagy begins with the formation of an autophagosome, a double membrane sphere that engulfs damaged cellular material. The physics of forming this sphere is extraordinary. A lipid bilayer membrane naturally wants to be flat. Bending it into a sphere requires energy, described by the Helfrich bending energy equation. The cost goes up with the square of the curvature. To form a sphere big enough to engulf a mitochondria, which can be several micrometers long, the membrane must be forced into a shape it inherently resists. The ATG proteins that Ohsumi identified do this mechanical work. They form a complex, the ATG12, ATG5, ATG16L1 complex, along with the LC3 lipidation system that acts like a molecular scaffold. They sense the flat membrane, recruit lipids to the growing phagophore, the expanding sheet, and drive the bending and enclosure that seals it into a sphere. The entire structure assembles in about 10 minutes. 10 minutes from a flat patch of membrane to a sealed double-walled sphere large enough to contain a damaged organelle. This isn't a metaphor for efficiency. It's Newtonian mechanics, friction and force playing out at the nanometer scale. For every 10 minutes you're in a fasted state with autophagy active, your cells are completing full construction and recycling cycles. It's a manufacturing and recycling operation running simultaneously, powered by the physical principle of membrane bending, all triggered by the simple absence of nutrients. And the cell doesn't just eat anything. It's precise. The most important discovery in recent autophagy research is selective autophagy, the cell's ability to identify specific damaged structures and target them for destruction while leaving healthy ones untouched. The clearest example is mitophagy, the selective clearance of damaged mitochondria. This is governed by the PINK1 Parkin system. PINK1 is a kinase that's constantly being imported into healthy mitochondria. Once inside, it's degraded. On the surface of a healthy high-voltage mitochondrion, PINK1 levels are virtually zero. It's invisible to the clearance machinery. But when a mitochondrion's inner membrane potential drops, when it falls below that 180 millivolt threshold that indicates dysfunction, the import machinery stops working. PINK1 can no longer enter. It accumulates on the outer membrane instead. That accumulated PINK1 then recruits Parkin, an E3 ubiquitin ligase. Parkin tags multiple proteins on the outer mitochondrial surface with ubiquitin molecules, creating a dense molecular flag. Autophagy receptors like P62 recognize these ubiquitin chains and recruit the growing autophagosome directly to that specific flagged mitochondrion. The cell identified a failing power plant by measuring its voltage, flagged it through a molecular cascade triggered by that electrical failure, and dispatched the recycling machinery precisely to that location. Think about the elegance. The cell doesn't need to chemically analyze the output of every mitochondrion. It doesn't need to run a complex audit of its DNA. It uses one simple continuous binary measurement, voltage. Above threshold, the mitochondrion is invisible. Below threshold, it's marked for death. The entire quality control decision for your cell's most critical power source runs on a single electrical parameter. In a constantly fed state with mTOR active, flagged mitochondria accumulate. The voltage surveillance works, the PINK1 marks them, but the autophagosome machinery is suppressed. The damaged mitochondria persist, producing less ATP, leaking more reactive oxygen species, accumulating further damage. Fasting releases the brake. The autophagosome machinery activates. The backlog of flagged mitochondria is cleared. Then new mitochondria are generated through a process called mitochondrial biogenesis, driven partly by PGC1 alpha, a master regulator activated by AMPK during fasting. They replace the ones that were removed. The mitochondrial population after a fast is younger, less damaged, and more efficient than the one that went in. The cell is not making a chemical judgment about health. It's making an electrical one. The physics of membrane potential, the same electrochemistry that drives nerve conduction, is the instrument the cell uses to decide which of its power plants to replace. The quality control is electrical. The measurement is voltage. The consequence is a renewal. Now here's the layer that changes the meaning of this entire process. Your mitochondria are not native to you. They are endosymbionts, ancient bacteria that took up residence inside a primitive cell about 1.5 billion years ago. They have their own DNA, their own ribosomes, which are more similar to bacterial ribosomes, and they still retain their own double membrane system. The inner membrane where the voltage is measured is their original bacterial membrane. Every cell in your body houses hundreds to thousands of these ancient bacterial guests. They supply about 90% of your ATP. Without them, complex life as we know it would be impossible. When your cell measures the voltage of a mitochondrion and flags it for recycling, it is managing its relationship with a symbiont older than all of animal life. It's using a voltage measurement, PINK1 accumulation, Parkin tagging, and autophagosome engulfment. All of this is a eukaryotic cell deciding which of its ancient bacterial residents are still functional and which need to be replaced. The inner membrane potential that pink one measures is generated by electrochemical physics, identical to what drives membrane potential in free-living bacteria today. The cell is checking whether its ancient residents are still running their original electrochemistry. The ones that aren't get recycled. Fasting, which triggers enhanced mitophagy, is accelerating a 1.5 billion-year-old quality control relationship between host and symbiont. The person fasting for 16 hours is not doing something new. They are activating a maintenance program that is older than multicellular life itself. This voltage-based bulk recycling system is one story, but there's another operating through entirely different physics, handling a different problem. This is chaperone-mediated autophagy, or CMA. Unlike macro-autophagy, which engulfs entire organelles, CMA works continuously, independent of fasting, degrading individual damaged proteins one molecule at a time. Chaperone protein, H70, recognizes damaged proteins carrying a specific amino acid sequence motif. It escorts them one by one to the lysosomal membrane. There, a receptor called LAMP2A unfolds the protein and threads it directly through the membrane into the lysosome for degradation. No autophagosome, no membrane bending, just molecular threading. CMA handles about 30% of all protein degradation under normal conditions. The recognition motif it targets is present in roughly 30% of all cytoplasmic proteins. The cell has, in a sense, pre-tagged nearly a third of its own protein inventory for monitoring. The surveillance is built into the protein's very structure. This matters profoundly as we age. CMA activity declines with age. LAMP2A levels on the lysosomal membrane drop, reducing the throughput of this continuous quality control system. The proteins that CMA would have caught and degraded individually start to accumulate. They aggregate. Those aggregates become targets for a bulk system called autophagy. But autophagy is a slower, more resource-intensive system for handling individual misfolded proteins than the molecular threading of CMA. It's the difference between picking up individual pieces of litter as they fall versus waiting for them to form a pile and then sending a truck. CMA is the continuous pickup. Autophagy is the truck. The accumulation of protein aggregates in aging cells, including the amyloid and tau implicated in neurodegenerative diseases, is partly a consequence of this declining CMA throughput. Fasting upregulates macroautophagy, the bulk system, but it doesn't restore CMA. The two pathways address different problems through different physics, decline at different rates, and respond to different interventions. To treat autophagy as a single process is to miss this essential distinction. Now, consider the brain in this fasted state. After about 18 to 24 hours without food, your liver's glycogen stores are depleted. It begins breaking down fat into molecules called ketone bodies, primarily Beta-Hydroxybutyrate. This crosses the blood-brain barrier and fuels your neurons directly. This is widely discussed. But what's almost never mentioned is that beta-hydroxybutyrate is not just fuel. It's a signaling molecule. It acts as a histone deacetylase inhibitor. It blocks the enzymes that remove acetyl groups from histone proteins, keeping your chromatin in a more open, transcriptionally active state. This has a profound effect. Among the genes that get expressed more robustly under beta-hydroxybutyrate is BDNF, brain-derived neurotrophic factor. BDNF is the master regulator of synaptic plasticity, learning, and the growth of new neurons in the hippocampus. It's the molecule that helps the brain rewire itself. The molecule that fuels your brain during fasting is simultaneously reprogramming your brain's gene expression toward growth, plasticity, and resilience. It's providing calories and performing a form of precision pharmacology at the same time through two completely different molecular mechanisms, all through one molecule. For the aging brain, where BDNF levels decline naturally, where synaptic plasticity decreases, where the hippocampus loses volume each year, this signal is profound. The fuel the fasted brain runs on is simultaneously the signal that drives the plasticity the aging brain is losing. There is no pharmaceutical that currently produces this combination, a metabolic fuel that is also a direct transcriptional reprogrammer. The mental clarity many people report during extended fasting, that sharpness after the initial fog, has a precise molecular basis. Beta-Hydroxybutyrate opening up the genome and increasing BDNF expression in neurons that are simultaneously running more efficiently on ketones than they were on glucose. The dual function fuel and reprogrammer is the finding that sits at the center of this whole story. The brain's response to fasting is not deprivation, it is activation. Now, a note on what is settled and what is not, because this matters. Ohsumi's work was in yeast. The extension to human clinical outcomes, exactly how many hours produce the best results, whether intermittent fasting prevents specific diseases, the optimal protocol is an active area of investigation. The mechanism is real. The optimal human protocol is still being defined. Both are true. Only one is settled. Dr. Walter Longo's work on the fasting mimicking diet at USC has produced the strongest human clinical data so far, showing improvements in biomarkers of aging and metabolic health. But his protocols are often more demanding than standard 16.8 intermittent fasting. The 16-hour window that popular culture promotes may or may not be sufficient for the level of autophagic induction the mechanism predicts. The threshold varies from person to person. It varies with metabolic health, age, and other factors. It hasn't been measured with the precision the mechanism deserves in large human trials. The person who tells you 16 hours will definitely clear your damaged mitochondria is extrapolating from yeast and mouse data. The person who tells you the mechanism isn't real is ignoring a Nobel Prize. And we return to the second law. It doesn't negotiate. Every ordered structure in the universe moves toward disorder unless energy is spent to maintain it. Your cells have been spending that energy since before you were born, maintaining membrane potentials, refolding proteins, measuring mitochondrial voltage, threading damaged molecules through lysosomal membranes one by one, bending lipid bilayers into spheres around failing organelles. Fasting doesn't introduce a new process. It shifts the balance of an existing one from building outward to repairing inward, from suppressing the recycling machinery to releasing it, from accumulating damage to clearing it. The mTOR switch flips, the autophagosomes form, the voltage surveillance clears the backlog. The damaged mitochondria are replaced with new ones. The misfolded proteins that escaped CMA are engulfed in bulk. The cell that was running maintenance in the background while prioritizing growth now prioritizes maintenance and suspends growth. The physics reverses direction. Now think about the modern eating pattern. A person who eats from 7 a.m. until 10 p.m. has a 15-hour feeding window. They have a 9-hour overnight fast. That 9 hours is never enough to flip the switch. The switch never flips. The autophagosomes never form at enhanced rates. The flagged mitochondria accumulate another day. The misfolded proteins that CMA missed remain in the cytoplasm. The cell runs another 24 hours in growth mode, accumulating the damage that repair mode would have cleared because the feeding window never closed long enough for the phase transition to occur. The food isn't harmful. The growth signal the food delivers holds the recycling brake in place, and the brake stays on as long as the food keeps arriving. This isn't an argument for any one fasting protocol. It's a description of what the physics requires. The threshold exists. The switch is binary. The cell cannot run enhanced autophagy while mTOR is active. And mTOR remains active as long as amino acids are present above a threshold concentration. The minimum duration required to cross that threshold in humans is somewhere between 12 and 16 hours. It varies. But the threshold is real, and the physics on either side of it is fundamentally different. The Nobel Prize wasn't awarded for discovering something exotic. It was awarded for describing the mechanism by which every cell in every body that has ever existed maintains itself against the most fundamental law in physics. Fasting doesn't damage your cells. It is the condition under which your cells do the work they were built to do. Fasting doesn't damage your cells. It's the condition where they do the work they were built for. That is the physics.